9 नंबर का हमारा चैप्टर बायोमोलिक्यूल्स। सो स्टार्ट करते हैं। देयर इज़ अ वाइड डाइवर्सिटी इन लिविंग ऑर्गेनिजम्स in our biosphere. Now a question that arises in our minds is, are all living organisms made of the same chemicals, that is elements and compounds? You have learned in chemistry how an elements analysis is performed. If we perform such an analysis on a plant tissue, animal tissues, or a microbial paste, we obtain a list of elements like carbon, hydrogen, oxygen, and several others. And list of elements like carbon, hydrogen, oxygen, and several others, and their respective content per unit mass of a living tissue. If the same analysis is performed on a piece of Earth's crust as an example of non living matter, we obtain a similar list. What are the differences between the two lists? In absolute terms, no such differences could be made out. All the elements present in a sample of Earth's crust are also present in a sample of living tissue. However, a closer examination reveals that the relative abundance of carbon and hydrogen with respect to other elements is higher in any living organisms than in Earth's crust. Table 9.1. How we analyze chemical and composition. We can continue asking in the same way, what type of organic compounds are found in living organisms? How does one go? How does one go about defining the answer? To get an answer, one has to perform a chemical analysis. We can take any living tissue, or vegetable, or piece of liver, etc., and grind it in trichloroacetic acid trichloroacetic acid using a not using a not using a mortar and a pestle. We obtain a thick slurry. If we were to thread this through a case of cloth or cotton, we would obtain two fractions. One is One is called the filtrate, or more technically, the acid soluble pool, and the second, the retentate, or the acid insoluble fraction. Scientists have found thousands of organic compounds in acid soluble pool. In higher classes, you will learn about how to analyze a living tissue sample and identify a particular organic compound. It will suffice to say here that the one extracts the compounds within subjects the extract to various separation techniques till one has separated a compound from all other compounds. In other words, one one isolates and purifies a compound. Analytical techniques, when applied to the compound, give techniques when applied to the compound give us an idea of the molecular formula and the probable structure of the compound. All the probable structure of all the carbon compounds that we get from living tissues can be called biomolecules. However, Living organisms have also got inorganic elements and compounds in and them. How do we know this? Slightly different but destructive experiment has to be done. One weighs one weighs a small amount of living tissue, say the leaf or cellulose leaf or layer, and it's this is called a weight and dry it. All the water evaporates, the remaining material gives dry weight. Now, if the tissue is fully burnt, all the carbon compounds are oxidized to gaseous form, carbon dioxide, water vapor, and the removed what the remaining is called ash. This ash contains This ash contains inorganic elements like calcium, magnesium, etc. Inorganic compounds like sulfate, phosphate, etc. are also seen in acid soluble fraction. Therefore, elemental analysis gives the elemental composition of living tissues in the form of hydrogen, oxygen, chlorine, carbon, etc. While analysis of for compounds gives an idea of the kind of organic figure 9.1 and inorganic constituents table 9.2 present present in living tissues from chemistry point of view. The one can identify functional groups like aldehyde, ketone, aromatic compounds, etc. But from a biological point of view, we shall classify them into amino acids, nucleotides, bases, fatty acids, etc. Amino acids are organic compounds containing an amino groups and an acid groups as substituents on the same carbon, that is the alpha carbon, hence they are called alpha amino acids. They are substituted methanes. There are four substituent groups occupying the four valency positions. These are hydrogen carboxyl group, amino group, and a variable group designed as R group based on the nature of R group. There are many amino acids, however, those which occur in proteins are only of 20 types. The R group is these proteinaceous amino acids could be a hydrogen. The amino acid is called glycine or methyl group, alanine, hydroxyl, methyl, serine, etc. Three of the 20 are shown in figure 9.1. The chemical and physical properties of amino acids are essential of amino carboxyl and are functional groups based on number of amino and carboxyl groups. There are acidic, for example, glutamic acid, basic lysine, and neutral valine amino acids. Similarly, there are aromatic amino acids, tyrosine, phenylalanine, tryptophan. A particular property of amino acids is the ionizable nature of NH2 and COOH group. Hence, its solutions of different pH. The structure of amino acids changes. B is called the zwitterionic form. Lipids are generally water insoluble. There could be simple fatty acids. The fatty acid has a carboxyl group attached to an R group. The R group could be methyl and ethyl, or higher number of CH2 CH2 groups, one carbon and 19 carbons. For example, palmitic acid has 16 carbon, including carboxyl carbon. Arachidonic acid has 20 carbons, including carboxyl carbon. Arachidonic acid has 20 carbon atoms, including the carboxyl carbon. Fatty acids fatty acids could be saturated atoms, including the carboxyl carbon. Fatty acids could be saturated without double bond or unsaturated with one or more carbon double bond. Another for simple lipid is glycerol, which is trihydroxypropane. Many lipids have both glycerol and fatty acids. Here the fatty acids are found. Here the fatty acids are found esterified with the glycerol. They can be then monoglycerides and diglycerides and triglycerides. These are also called fats and oils based on melting point. Oils have lower melting point, for example, gingelly oil, and hence remain as the oil is winters. Can you identify a fat from market? Some lipids as oil in winters. Can you identify fat from the market? Some lipids have phosphorus and the phosphorylated organic compound in them. These are phospholipids. They are found in cell membrane. Lecithin is one example. Some tissues, especially the neutral tissues, have lipids with more complex structures. Living organisms have a thin number of carbon compounds in which heterocyclic rings can be found. Some of these are nitrogen bases: adenine, guanine, cytosine, uracil, and thymine. When found attached to a sugar, and they are called nucleosides. If a phosphate group is also found esterified to the sugar, they are called nucleotides. Adenosine, guanosine, thymidine, uridine, and cytidine are nucleosides. Adenylic acid, thymidalic acid, guanylic acid, uracil acid, and cytidinic acid are nucleotides. Nucleic acids like DNA and RNA consists of nucleotides only. DNA and RNA function as genetic material. Sugars, amino acids, fats, and oils, nitrogen bases, nucleotides, nucleosides, farm, primary and secondary metabolites. The most exciting aspect of chemistry deals with isolating on thousands of compounds, small and big, from living organisms, determining their structure and, if possible, synthesizing them. If one were to make a list of biomolecules, such a list would have thousands of organic compounds, including amino acids, sugars, etc., for reasons that are given in section 9.10. We can call these biomolecules as metabolites in some animal tissues. One notices the presence of all such categories of compounds shown in figure 9.1. These are called primary metabolites. However, when one analyzes plants, fungal, and microbial cells, one curve would see thousands of compounds other than these are called primary metabolites. Example: alkaloids, flavonoids, rubber, essential oils, antibiotics, colored pigments, scents, gums, spices. These are called secondary metabolites, table 9.3, while primary metabolites have identifiable functions and play known roles in forming normal physiological processes. We do not at the moment understand the role or functions of all the secondary metabolites in host organisms. However, many of them are useful to human welfare. For example, rubber, drugs, spices, scents, and pigments. Some secondary metabolites have ecological importance in the latter chapters. Have years, you will learn more about this. Biomacromolecules. There is one feature common to all those compounds found in the acid soluble pool. They have molecular weights ranging from 18 to around 800 daltons approximately. The acid insoluble fraction have only four types of organic compounds, that is proteins, nucleic acids, polysaccharides, and lipids. These classes of compounds, with the exception of lipids, have molecular weights in the range of 10,000 daltons and above. And for this very reasons, biomolecules, that is chemical compounds found in living organisms, are of Dalton and are usually referred to as micromolecules or simply biomolecules, while those which are found in the acids insoluble fraction are called macromolecules and biomacromolecules. The molecules in the insoluble fraction, with the exception of lipids, are polymeric substances. Then vary why do lipids whose molecular weights do not exceed 800 Dalton. Some come under acid insoluble fraction, that is macromolecular fraction. Lipids are indeed small molecular weights compounds and are present not only as such but also arranged into structures like cell membrane and other membranes. When we grind a tissue, we are disrupting the cell structure. Cell membrane and other membranes are broken into pieces and form vesicles which are not water soluble. Therefore these membrane fragments in the form of vesicles get separated along with the acid insoluble pool, and hence in the macromolecular fraction lipids are not strictly macromolecules. The acid soluble pool represents roughly the cytoplasmic composition. The macromolecules from cytoplasm and organelles become the acid insoluble fraction. Together they present and together they represent the entire chemical composition of living tissues or organisms. In summary, if we represent the chemical composition of living tissues from abundance point of view and arrange them classwise, we observe that water is the most abundant chemical in living organism. Table 0.4 9.4. Proteins are polypeptides. They are linear chains of amino acids linked by peptide bonds as shown in figure 9.3. Each protein is a polymer of amino acids, as there are 20 types of amino acids. For example, alanine, cysteine, proline, tryptophan, lysine, etc. A protein is a heteropolymer and are not homopolymer. A homopolymer has only one type of monomer repeating n number of times this information about amino acid content is important as later in your nutrition lessons you will learn that certain amino acids are essential for your our health and to be suppli- they have to be supplied through our diet. Hence, dietary proteins are the source of essential oils, amino acids. Therefore, amino acids can be essential or non-essential. The latter are those which our body can make, while we get essential amino acids through our diet. Food proteins carry out many functions in living organisms. Some transport nutrients across cell membrane, some flight infectious organisms, some are hormones, some are enzymes, etc. Table 9.5. Collagen is the most abundant protein in animal world and for ribulose, biphosphate, carboxylase, oxygenase, ribisco is the most abundant protein in the whole of the biosphere. Polysaccharides, the acid insoluble pellet, also has polysaccharides. Carbohydrates has another class of macromolecules. Polysaccharides are long chains of sugars. They are threads, literally cotton thread, containing different monosaccharides as building blocks. For example, cellulose is a polymeric polysaccharides consisting of only one type of monosaccharides, that is glucose. Cellulose is a homopolymer. Starch is a variant of this but present as a storehouse of energy in plant tissues. Animals have another variant called glycogen. Inulin is a polymer of fructose in a polysaccharide chain, say glycogen. The right hand is called the reducing, shown in the form of carbon figure 9.2. Starch forms helical secondary structures. In fact, starch can hold I2 molecules in the helical portion. The starch I2 is blue in color. Cellulose does not contain complex helices, and hence cannot be hold I2. Plant cells, plant cell walls are made of cellulose. Paper made of from plant pulp and cotton fiber is cellulosic. There are more complex polysaccharides in nature. They have as building blocks amino sugars and chemically modified sugars. For example, glucosamine, N-acetyl galactosamine, etc. Exoskeletons are of arthropods, for example, have a complex polysaccharide called as chitin. These complex polysaccharides are mostly homopolymers. Nucleic acids, the other type of macromolecules that one would find in the acid insoluble fraction of any living tissues, is the nucleic acid. These are polynucleotides together with polysaccharides and polypeptides. These comprise the true macromolecules, a fraction of any living tissues or cell. For nucleic acids, the building block is a nucleotide. Then nucleotide has three chemically distinct components. One is heterocyclic compound, and the second of monosaccharide, and the third phosphoric acid or phosphate. As you notice in figure 9.1, the heterocyclic compounds in nucleic acids are the nitrogenous bases named adenine, guanine, uracil, cytosine, and thymine. Adenine and guanine are substituted purines, while the rest of substituted pyrimidines, and the skeleton heterocyclic ring is called purine and pyrimidine, respectively. The sugar compound is the sugar found in deoxyribose. A nucleic acid containing the deoxyribose is called deoxyribonucleic acid DNA, while that which contains ribose is called ribonucleic acid RNA. Structure of proteins. Proteins, as mentioned earlier, are heteropolymers containing strings of amino acids. Structure of molecules means different things in different contexts. In organic chemistry, the structure invariably refers to the molecular formulae: sodium chloride, magnesium chloride, etc. Organic Organic chemists always write a two-dimensional view of the molecules while representing the structure of the molecules. Example: benzene, naphthalene, etc. Physicists conjure of the three-dimensional views of molecular structures, while biologists describe the protein structure as at four levels: the sequence of amino acids, etc. That is the positional information in a protein, which is the first amino acid, which is second, and so on, is called the primary structures. Figure 9.31a of the protein is called imagined as a line left hand represented by the first amino acid and the right hand represented by the last amino acid. The first amino acid is also called as N-terminal amino acid. The terminal amino acid, a protein thread, does not exist throughout as an extended rigid rod. The threads is folded in the form of helix, similar to a revolving staircase. Of course, only some proportions of the protein threads are arranged in the form of a helix, and proteins have only right-handed helices are observed. Other regions of the protein thread are folded into other forms in what is called the secondary structures. Figure 9.3b. In addition, the long protein chain is also folded upon itself like a hollow woolen ball, giving rise to the tertiary structure like YCC. This gives us a three-dimensional view of a protein. Tertiary structure is absolutely necessary for the many biological activities of proteins. Some proteins are an assembly of more than one polypeptide or subunits. The manner in which these individual folded polypeptides or subunits are arranged with respect to each other. The example: linear string of spheres arranged of one upon each other in the form of cube or plate, etc. is the architecture of a protein, otherwise called the quaternary structure of a protein. For example, figure 9.3d. Adult human hemoglobin consists of four subunits. Two of these are identical to each other, hence two subunits of alpha type and two subunits of beta type together constitute the human hemoglobin. Enzymes. Almost all enzymes are proteins. There are some nucleic acids that behave like enzymes. These are called ribozymes. One can depict an enzyme by a line diagram. An enzyme, like an protein, has a primary structure, that is amino acid sequence of a protein of the protein. An enzyme, like any protein, has a secondary and the tertiary structure. When you look at a tertiary structure figure at one, you will notice that the black backbone of protein chain folds upon itself. The chain crisscross crisscrosses itself, and hence many crevices or pockets are made. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. Thus, enzymes, through their active site, catalyze reactions at a high rate. Enzyme catalysts differ from inorganic catalysts in many ways, but one major difference needs mention. Inorganic catalysis work efficiently at high temperatures and high pressures, while enzymes get damaged at high temperatures, say above 40 degrees Celsius. However, enzymes are isolated from organisms who normally say above 40 degrees Celsius. However, enzymes isolated from organisms who normally live under extremely high temperatures. Example: hot winds and sulfur springs are stable and retain their catalytic power even at the high temperatures up to 80 to 90 degrees Celsius. Thermal stability is thus an important quality of such enzymes isolated from thermophilic organisms. Chemical reactions. How do we understand these enzymes? Let us first understand a chemical reaction. Chemical compounds undergo two types of changes. A changes. Physical change simply refers to a change in shape without breaking of bonds. Now, this is a physical process. Another physical process is a change in state of matter when ice melts into water or when water becomes a vapor. These are also physical processes. However, when bonds are broken and new bonds are formed during transformation, this will be called the chemical reaction. For example, BaOH2H2SO4 then gives BaSO4 and 2 times water. Is an inorganic chemical reaction. Similarly, hydrolysis of starch into glucose is an organic chemical reaction. Rate of a physical chemical process refers to amount of product formed per unit time. It can be expressed as rate delta Pa upon delta T. Rate can also be called velocity if the direction is specified. Rates of physical and chemical processes are influenced by temperature, among other factors. The general rule of thumb is that the rate doubles and or decreases by half rates vastly higher than that of uncatalyzed ones. When enzyme catalyzed reactions are observed, the rate will be higher than the same but uncatalyzed reaction. For example, CO2, carbon dioxide plus water, carbonic analysis and hydrogen presence, and H2SO for carbonic acid. In the absence of any enzyme, this reaction is very slow, with about 200 molecules of H2CO3 being formed in an hour. However, by using the enzyme present within the cytoplasm called carbonic anhydrase, the reaction speeds dramatically with about 6 lakh molecules being formed every second. The enzyme has accelerated the reaction rate by about 10 million times. The power of enzymes is incredible indeed. There are thousands of types of enzymes, each catalyzing a unique chemical or metabolic reaction, a multi-step chemical reaction. When each of the steps are catalyzed by the same enzyme, complex or different enzymes, it's called a metabolic pathway. For example, glucose pyruvic acid, two times pyruvic acid, is actually a metabolic pathway in which glucose becomes pyruvic acid through 10 different enzyme catalyzed metabolic reactions. When you study respiration in chapter 12, you will study these reactions. At this stage, you should know that this very metabolic pathway with one or two positions additional reactions gives rise to a very variety of metabolic and products in our skeleton muscles. Under under anaerobic conditions, lactic acid is formed. Under normal aerobic conditions, pyruvic acid is formed in yeast during fermentation. The same pathway leads to the production of ethanol alcohol. Hence, in different conditions, different products are possible. How do enzymes bring about such high rates of chemical conversions? To understand this, we should study enzymes a little more. We have already understood the idea of an active site, the chemical or metabolic conversion refers to a reaction. The chemical which is converted into a product is called a substrate. Hence, enzymes is proteins which three dimensional structures includes the active site convert a substrate into a product symbolically. This can be depicted as it is now understood that the substrate, yes, has to bind the enzyme at its active site within a given cleft or pocket. The substrate has to diffuse towards the active site. There is thus it's a obligatory formation of an ES complex. E stands for enzymes. This complex formation is a transient phenomena during the state where substrate is bound to the enzyme active site. The new substructure of the substrate called the transition state structure is formed very soon after the expected bond making is completed. The product is released from the active site. The other words, the structure of substrate gets transformed into the structure of products. The pathway of this transformation must go through goes through the so-called transition state structure. There could be many more altered structural states between the stable substrate substrate and the product implicit in this statement in the is the fact that all other intermediate structural states are unstable. Stability is something relates to energy status of the something relates to energy status of the molecules or the structure. Hence, when we look now at this pictorially through a graph, it looks like something as the in figure 9.4. The y-axis represents the essential energy content. The x-axis represents the progressional of the structural transformation states through the transition state. Now you would notice two things: the energy level difference between the S and P. If P is at a lower level and then yes, the reaction is an exothermic reaction. One need not supply energy by hand heating in order to form the product. However, whether it is an exothermic spontaneous reaction or an endothermic or energy requiring reaction, Yes has to go through the much higher energy state of transition state. The difference in average energy content of yes from that of this transition state is called activation energy. Enzymes eventually bring down this energy barrier, making the transition of S and P more easy. Nature of enzyme action: each enzyme has a substrate bind, binding site in its molecules, so that the highly reactive enzyme substrate complex is productive is produced. This complex is short-lived and dissociates into its product and unchanged enzymes with an intermediate formation of the enzyme product complex. The formation of ES complex is essential for catalysis. The catalytic cycle of an enzyme action can be described in the following steps. First, the substrate binds to the active site of the enzyme, leading into the active site. The binding second, the binding site, the binding of the substrate induces the enzyme to alternate to alter its shape, fitting more tightly around the substrate. The active site third, the active site of enzyme now in close proximity of the substrate breaks the chemical bonds of the substrate and the new enzyme product complex is formed. The enzyme releases the products of the reaction and the free enzyme is ready to bind to another molecule of the substrate and run through the catalytic cycle once again. Factors affecting enzyme activity. The activity of an enzyme can be affected by a change in the conditions which can alter the tertiary structure of the protein. These include temperature, pH range, pH change in substrate, concentration or binding of specific chemicals that regulate its activity, temperature, and pH. Enzymes generally function in a narrow range of temperature and pH. Figure 9.5. Each enzyme shows its highest activity at a particular temperature and pH called the optimum temperature and optimum pH. Activity declines both below and above the optimum value. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity. because proteins are denatured by heat. Concentration of substrate. With the increase in substrate concentration, the velocity of enzymatic reaction rises at first. The reaction ultimately reaches a maximum velocity Vmax, which is not exceeded by any further rise in concentration of the substrate. And this is because of the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules there are no free enzyme molecules to bind with the additional substrate molecules. Figure 9.5, the activity of an enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When binding of the chemical shuts off enzyme activity, the processes is called inhibition, and the chemical is called an inhibitor. When the inhibitor closely resembles the substrate in its molecular structure and inhibits the activity of the enzyme, it is known as competitive inhibitor. Due to its close structural similarity with the substrate, the inhibitor competes with the substrate for the substrate binding site of the enzyme. Consequently, the substrate cannot bind, and as a result, the enzyme action declines. Example: inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in structure. Such competitive inhibitors are often used in the control of bacterial pathogens. Classification and nomenclature of enzymes. Thousands of enzymes have been discovered, isolated, and studied. Most of these enzymes have been classified into different groups based on the types of reactions they catalyze. Enzymes are divided into six classes, each with 4 to 13 subclasses, and named accordingly by a four-digit number. Oxido reductase or dehydrogenases enzymes which catalyze oxidase and between two substrates S and S. Example: reduced oxidized S, reduced X oxidized S oxidized S, reduced transferases enzymes catalyzing a transfer of a group G other than hydrogen between a pair of substrate S and S. Example: hydrolysis enzymes catalyzing hydrolysis of ether, peptide, glycosidic acid, CC, carbon halide, and PN bonds. Lysis enzymes that catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving double bonds. Isomerases includes all enzymes catalyzing interconversion of optical, geometric or positional isomers. Ligases enzymes catalyzing the linking together of two compounds. Example: lyase enzymes which catalyze joining of carbon oxygen, carbon sulfur, carbon nitrogen, phosphorus oxygen, etc. bonds. Cofactors enzymes are compounds of one or several polypeptide chains. However, there are a number of cases in which non-protein constituents called cofactors are bound to the enzyme to make the enzyme catalytically active. In these instances, the protein portion of the enzymes is called the apoenzymes. The three kinds of cofactors may be identified: prosthetic groups, coenzymes, and metal ions. Prosthetic groups are organic compounds and are distinguished from other factors cofactors in that they are tightly bound to the coenzyme. For example, in peroxidases and catalase, which catalyze the breakdown of hydrogen peroxide to water and oxygen. Heme is the prosthetic group, and it is a part of the active site of the enzyme. The coenzymes are also organic compounds, but their association with the apoenzymes is not transient, usually occurring during the course of catalysis. Furthermore, the coenzymes serve as cofactors in a number of different enzyme catalyzed reactions. The chemical opponent of many coenzymes are vitamins. Example: coenzyme nicotinamide, adenine, and dinucleotide, NAD, and NADP contain vitamin niacin. The number of enzymes require metal ions for their activity, which form coordination bonds with side chains at the active site, and at the same time from one or more coordination bonds with the substrate. Example: zinc is a cofactor for the proteolytic enzyme carbonylase. Catalytic activity is lost when the cofactor is removed from the enzyme, which testifies that they play a crucial role in the catalytic activity of the enzyme. तो यहाँ पर बायोमोलिक्यूल्स चैप्टर खत्म होता है। ये रही उसकी समरी। तो यहाँ पर बायोमोलिक्यूल्स चैप्टर खत्म होता है। सो हमने इसमें देखा how to analyze chemical composition in charts, then sugar, then amino acid, fats and oils, nitrogen bases, nucleotides, nucleosides, primary and secondary metabolites, macromolecules, proteins, polysaccharides, nucleic acids, structure of proteins, enzymes, chemical reactions. How do enzymes bring about such high rates of chemical conversions? Nature of enzyme action, factors affecting enzyme activity, classification and nomenclature of enzymes, then summary. So यहाँ पर बायोमोलिक्यूल्स चैप्टर खत्म होता है। थैंक यू सो मच फॉर वॉचिंग। प्लीज़ लाइक शेयर एंड सब्सक्राइब।